Okay, so I know we talked about capillary pressures as we were moving from aorta to vena cava, but we need to take a look at them here because this helps set you up for the kidney chapter, which is kind of a challenge. So knowing these pressures and this type of exchange will lay a foundation for you to be successful there. There are two kinds of pressures that exist in your capillaries. There's a hydrostatic pressure, which is the pressure of water, kind of like if you had a water balloon, that's the pressure the water exerts from the inside out of the balloon. And there's also something called colloid osmotic. So a colloid is just a substance in water that attracts water. Basically, these are solutes. That's what a colloid is. So here, down in our capillary below in the picture, the hydrostatic pressure, the pressure of water, pushes water and solutes out of the capillary to the surrounding tissues. The colloid osmotic pressure attracts that water back. There are two kinds of exchange, and you can see them here on this diagram. There's filtration, which is the blue arrow pushing solutes and water from the capillary to the surrounding tissues. And then there's reabsorption, which is mostly just water, and that's the green arrow, and it's the attraction back into the capillary of fluids. I like this picture down here because it's got a capillary, but it's also got the tissue cells surrounding it, and then an open space between the capillary and the tissue cells, which would be the interstitial fluids. Filtration is shown over on the left side of this diagram because the arrows are showing that substances are moving from the capillary to the interstitial fluid and then into the intracellular fluid of the tissue cells. So filtration is moving plasma, things that are plasma, to the interstitial fluids, right? So it pushes water out, but most of the solids stay in. Some solids can escape, like glucose and amino acids. And the force that's driving this is the hydrostatic pressure of water. Reabsorption is obviously, well, it's kind of the opposite of this. It's shown by the arrows over on the right side of this diagram because substances are moving from the interstitial fluids into the plasma. So this is when mainly fluid moves back into the capillary. And remember that water follows salt. So the fluid, being water, will be attracted to the solutes that are in the capillary. And this, of course, because we're talking about solutes, this would be an effect of the colloid osmotic pressure. This is the diagram in your book, which is not as pretty as the other diagrams. But what it's showing you here, let's read it from left to right. It's showing you that blood from an arteriole enters into a capillary. And you know that it enters into a capillary bed, but we're going to take this as a, abbreviation of a capillary bed. So blood enters from the arteriole and it gets pushed out of the capillary by filtration. And that's the big gray arrow that's leaving the capillary. And then it gets reabsorbed into the capillary by reabsorption. What this creates is a net filtration pressure. You have the pressure of filtration, the pressure of reabsorption, and the difference between those two is the filtration pressure, because you guys already know that there is more filtration than reabsorption. And this is true. There is always more fluids leaving the capillary than being reabsorbed into the capillary. And what takes that difference are the lymphatics. Lymphatics, which is a drainage system, are able to take larger substances than the capillaries. So things like white blood cells and maybe large hormones and enormous proteins that are created by your tissue cells. So lymphatics take that difference and they dump it back into circulation at the subclavian vein. So why does this matter, filtration and reabsorption? This is about tissue perfusion. This is about how many of those things, oxygen and glucose and water, enter into your tissues, leave the capillary, and enter into your interstitial fluids. Why does this matter? Well, you want good tissue perfusion. You want to make sure that there's an exchange between your interstitial fluids and the plasma and the things that are floating around in your blood vessels, right? Good perfusion means that you have good distribution of oxygen and glucose. You have good distribution of oxygen and carbon dioxide. You have good distribution of those hormones that have been secreted into your circulatory system by your endocrine cells and tissues. You also want to make sure that you're flushing toxins and pathogens along with your white blood cells into the lymphatics, because these are the things that cannot enter back into your capillaries, and you got to remove them from your tissues. Another issue that comes up that many students are familiar with and is a product of too much filtration and not enough reabsorption is edema. Edema can happen from a couple different things. It's usually from increased permeability of vessels or from too few colloids or proteins in the capillaries. So the colloids can't reclaim the water through reabsorption, and the water stays in the tissues and they swell, causing edema. There is a clinical note in your book on page 744, and it explains edema. It's really good. So the definition, you may want to say to me that edema is when your cells swell. No, it's when the spaces between your cells swell or accumulate interstitial fluid. Bruising can do this by damaging the capillary, making it more permeable. Starvation can do this by depleting your proteins in your capillary, making it difficult to attract water from the interstitial fluids. Or just simply if you have too much pressure, right? Your blood pressure is enormous and it keeps pushing water and colloids from the capillary to the interstitial fluids simply by hydrostatic pressure. What this will end up causing is peripheral congestion. And if you have a problem with the right side of your heart, a valve, it'll end up causing peripheral congestion, and edema in the end just keeps backing up until you're backed up through the whole